Okay. For edits, will be right now. Hi everyone, this is group theta-hats or overachieving theta-hats, and today we're going to be presenting on our project of exploring cell assemblies on large data sets. So we got this data set from Stringer et al. 2019, and this quick paper, just a very quick summary. They had 8 neuropixel probes totalizing about 10,000 neurons with calcium imaging. So we grabbed that data alongside spontaneous mouse behavioral, looking at eigenfaces. And our goal with that was to basically identify potential cell assemblies during those specific mouse behaviors or during stimulus-driven events.
One of the main methods we wanted to use for cell assembly detection was an unsupervised methodology called non-matrix factorization, or NMF. And this allowed us to pull out assemblies that were given by the user. So we would ask for, in this example, 10 factors. So we had 10 assemblies, and the NMF would give us a neuron factor and a temporal factor. So these group of neurons are somehow co-activated together in neuron factor 1, and they have this temporal factor and temporal factor 1. Unfortunately, we were unable to kind of determine how many factors to ask for. We looked at reconstruction error, but that seemed to just decrease over the number of factors, and it was unclear. But for the moment, we asked for anywhere from between 3 or 10 factors. Some things then to consider, just very simple statistics, was looking at the overlap of factors. So taking the top 10% of the weights in the neuron factor, seeing how many are presented in the other factors, and seeing if maybe some factors are represented by the same neurons or not. From this data set, we see that more or less it is not represented highly, at least. Maybe 20 to 40% of neurons are in other factors. And then we also looked at the temporal factors. We just did a simple Pearson correlation between each temporal factor and tried to find if there was any correlation in time. And mostly what we see is that most of the factors are negatively correlated. There are some that are positively correlated, but it will require further investigation to understand the meaning of these correlations.
Okay, so here I set the factor number to 3 just because it's easy to visualize, and we wanted to visualize each neuron's weight for each of the factors in the physical space. And the assumption is that neurons with big contributions to the same factor may be close to each other, meaning they're likely to belong to the same assembly. Here is a spatial representation of all neurons with colors corresponding to the depth of the neuron in the cortex. If I project them onto the XY plane, you see a 2D histogram like this. So it's clearly not evenly sampled, probably because of the blood vessels on the cortex. To visualize neurons that belong to the 3 assemblies, I plotted I plot those with weight bigger than 90% of the weight distribution for each factor. And the darker the color is, the higher the weight is for that factor. So similarly, we can project them onto the XY plane, and we see the pattern like this, showing they're more concentrated in space. It suggests that these cell assemblies tend to be more clustered in space. Okay, so we want to consider each of the factor as a subset of neurons and visualize the response of these groups. Here I plot the temporal trace of the entire population by averaging the neural response. So you can see it's pretty random. And if you plot the temporal trace for each factor, you see some kind of pattern in there. And similarly, if you plot the behavior data, here I use 3 features. One is running speed, and one is pupil size. The other one is the first principal component of the face movement. And then you can do a correlation between these factor responses to the behavior data. So here I just list all the correlation coefficients, and you can tell that factor 2 correlates with all of these behavior data pretty well, and also factor 1 correlates with the face movement. Okay, so next we wanted to visualize the population response in this 3D space defined by our 3 factors. So the position of each dot XYZ represents a certain time point, the response of each of the three factors. So here I just separated these dots by looking at the behavior data. So the blue dots means the time points where the mouse has a high running speed, and the red dots are the mice has a low running speed. So the high and low threshold is defined by the 50 percentile of the running speed distribution. So you can see, just based on the behavior data, the neuron response is pretty clustered and well separated in this 3D dimension. And it's the same for pupil size and also the first principal component for face movement. So I think it's pretty interesting because we use a completely unsupervised matrix decomposition method and represented this 10,000 neurons response in this 3D space, and also found the corresponding different behavior states can be separated in this space. Okay.
So while we weren't able to use the unsupervised methods to deduce the exact number of factors that we wanted to use, what we ended up doing was also trying different regression models to see if we can find distinct assemblies that are responding to a behavior event. In this case, it was the pupil size that we were trying to measure. So we tried to do a lasso regression because we thought that, you know, there'd be sparse coding of the predicted pupil size. That was a hypothesis. And so lasso regression would let us to set certain weights on certain neurons to 0 to sort of exclude them, and then take the neurons that didn't have weights equal to 0 and sort of call them as like a assembly and see how they corresponded to pupil size. But we were wondering, should we use a lasso regression or a lasso regression where we set the weights to be forced to be positive? Because if you think about it, neuronal activity, we might expect it to correlate with pupil activity, but negatively correlate with pupil activity, it might not make much sense. So we tested both, and looking at the models, it looks like the lasso regression performs a little bit better than the lasso regression when it was forced to be positive, but they're almost about the same. So the question is, which is the better model for our hypothesis? And so, what we ended up, going to the next slide. We ended up comparing these two models, and it turns out that depending on the hypothesis, you can get two different sort of analyses. And when you look at the lasso positive assembly, you can see that it would correlate much more positively and much more reliably with pupil area than would the pupil area with just the lasso assembly, even though the lasso assembly had a better predictive factor. And you can see just by looking at these plots on the top left that indeed the lasso positive assembly seems to correlate better with the pupil area than the lasso regression. So that told us that you know, just because a model is better or predicts something better doesn't necessarily mean it's more useful depending on the research question we want to ask.
We also have a project that, well, we have another idea we're working on, and it is to use a mark point process along with a Bayesian model to predict the pupil size based on the calcium imaging data. The way this, in order to decode the pupil size based on the calcium data. So the way this model would work is that a portion of our data would be our encoding model, in which we create distributions of the marks, which are the amplitudes of the spikes. And at each time point, use the marks, and at each time point, look at the states that are observed during those times and create a distribution of how likely it is to observe a spike of a particular amplitude at that time, and then relate it to the pupil size. So by using the spiking activity, we hope to create this, we hope to extract information from the pupil size or predict the pupil size. So essentially, yes, we get the calcium activity, They're going to have spikes amplitudes that are associated with the spikes, and using that information, we plan to decode the pupil size.
So in conclusion, the main things that we learned from this project was that the NMF did give us, we used an unsupervised method to try to get out the assemblies from the NMF, but it was very difficult to choose a number of factors to properly understand the network. And we believe this is probably just due to the nature of the very large data set that we had. The assemblies revealed interesting spatial clustering patterns, and some ensemble activities showed correlations with behavior. And we also saw that supervised learning efficiently identified assemblies with correlated activity to behavioral measures like the pupil area of interest. But we also understood that just because the model is fitting best, it did not necessarily mean that it would be most useful for interpretation depending on what research question we had. And with that, we thank you for listening. And we'd also like to thank our TA, Anna, and our mentor, Surya, for helping us out.